All right, hi everyone. My name is Cheng. So Lizzie and Malvika just gave some really great talks, and they talked a lot about kind of sequencing DNA to find potentially pathogenic variants. So my talk is going to be a little bit different. I'm going to be talking about sequencing RNA instead to find pathogenic variants. And so I'm currently a first-year PhD student in genome sciences. I'm also in the MD PhD program, and my mentor, Andrew Srigachis, is also a MD PhD investigator. So for those of you who are interested in like physician scientists and kind of medicine, hopefully my talk will give you a little bit of background about kind of what life is like for a medical geneticist. Okay, so today I'm going to talk about how I am trying to unravel genetic mysteries using long-read transcript sequencing. And I want to start off by providing the motivation for my talk. So imagine that we are in a medical genetics clinic, and we have a patient that comes in, and this patient has a genetic condition. And so it's our job to identify the genetic cause for this patient. So this patient has a genetic disorder, but we don't know the genetic cause. And we want to be able to find the exact genetic mutation that is causing this disease. And the reason why we want to do this is because this is really important for the patient, because we can, for example, we can predict how likely is a patient going to have kids that are going to have the same condition. We can also help predict whether or not the patient's extended relatives might have this condition as well. So this is really quite important. So we have this patient and we want to find the genetic cause. So what we do is we take cells from this patient, we extract DNA and then RNA, and then we sequence the DNA and RNA. And when we get a patient's cell, we actually get the patient's entire genome. And so here I represented a patient's genome as an apple. And so the human genome is huge. It's 3 billion base pairs, like Stephen said earlier. So this is a lot of data to work with. So as a result, a lot of times we don't end up sequencing the entire genome, or we sequence the entire genome, but we don't end up looking at the entire genome. Instead, we only look at what's called the exome, which is the coding parts of the genome. This is a much smaller part of the genome. But the problem is, if we're only looking at the exome, we're essentially missing out on the non-coding stuff. So the non-coding parts of our genome is essentially this huge mystery that we're not able to use to find pathogenic variants. And so the focus of my PhD is on getting a bigger slice of the apple, like coming up with a better approach to find pathogenic variants in the non-coding genome. So what are some examples of pathogenic non-coding variants? So non-coding variants can cause disease via a lot of different mechanisms. So as you can see here, and this is like super intimidating. So the main point that I want to get across from this slide is just that non-coding pathogenic variants can fall into two major categories: splicing variants in the blue and then regulatory variants in the red. So the big question of my PhD is I want to figure out how can we discover these variants. And my talk today will focus primarily just on splicing. And here's the outline of the talk I'll be using. So let's just start off by talking about what splicing is. So here we have DNA, and DNA is transcribed into RNA. And splicing is this really important step that allows us to go from premature mRNA to mature mRNA, and then mRNA is then translated to proteins. And a key takeaway that I want to talk about on this slide is that there are many places where we can introduce a mutation, and those mutations might cause a splicing defect. But if we just sequence the DNA, then we're not really able to say whether splicing is affected or not. So as a result, we also have to sequence the RNA. So this is why it's so important in my project to sequence the RNA. So we can come back to the slide and we can update it by saying that we're going to use RNA sequencing as our method to find pathogenic splicing variants. And so there are actually many ways of doing RNA sequencing. So there's something called short-read RNA sequencing. So we have the gene here. We have the mRNA transcripts, also known as isoforms here. And what a lot of people do is we first fragment the mRNA into smaller pieces, and then we sequence these smaller pieces, and then we have to kind of piece each small pieces back to form the entire isoform. So that's one way to do it. That's a more typically a cheaper way to do it, which is why it's so prevalent. But I think a slightly better way to do it is actually to use long-read sequencing. So instead of fragmenting the mRNA and sequencing the fragments, we actually just sequence the entire isoform in a single read. So this way, we don't have to go through the process of puzzling and then putting back each piece. We can just get an entire read representing an entire isoform. So in my project, because long read sequencing is so much better, this is the type of sequencing that I'm going to be doing. Okay, so we can come back and then slightly update it and say that we're going to use long read RNA sequencing. So at this point in my talk, I described how we want to find pathogenic splicing variants because this can help us solve the genetic cause for our patients. And I talked about how we're going to use long read RNA sequencing as our method of choice. So now I want to introduce what are we exactly looking for when we're sequencing a patient's RNA. So for my PhD, when I sequence a patient's RNA, the very specific thing that I'm looking for is something called nonsense-mediated decay. And nonsense-mediated decay is basically when mRNA becomes degraded because it contains a premature stop codon. So the main point here is that nonsense-mediated decay is a sign of, is when transcripts are actually degraded, and it's a sign of a pathogenic splicing variant. And an important point that I want to make here is that there are treatments called antisense oligonucleotide therapies that are currently being designed to target nonsense-mediated decay specifically. So being able to discover nonsense-mediated decay is actually really critical if we want to come up with treatments for our patients. And in order for us to find nonsense-mediated decay, we do RNA sequencing after treating patients with this chemical called cyclohexamide. This is a little bit too technical, so I'm not going to go into it, but just trust me that, that it works. If we treat it with cyclohexamide, we're going to find nonsense-mediated decay. So I just explained our general experimental design. And so now I want to show how we actually apply this in an actual patient. So the patient that I want to present is a 42-year-old who came to the clinic with symptoms of Charcot-Marie-Tooth disease. So this is a genetic neuromuscular disorder, and one of the key characteristics is a highly arched feet, as you can see here in this picture. In addition to highly arched feet, they also have a lot of other nerve deficits, so sensory as well as motor deficits as well. And so what we want to do is we want to provide a definitive diagnosis to this patient. So what we did was we did genetic testing. So we looked at all the coding parts in this patient genome, but we weren't able to find any pathogenic variants when we just looked at the coding DNA. So the next step we did was we actually did RNA sequencing. And because short read RNA sequencing is cheaper and easier to do, that's the first thing that we actually did. And so to be specific, actually, our collaborators at UCLA actually did the short read sequencing. And when we did short read sequencing, we found that this gene MFN2 has lower expression. And this is really great news because MFN2 is actually already associated with Charcot-Marie-Tooth disease. So at this point, we know that this patient has symptoms of Charcot-Marie-Tooth disease. And we know that MFN2 is likely the genetic cause for this patient, but we don't actually know the mechanism. So we don't know if there is a splicing variant that is causing nonsense-mediated decay in this patient. And so this is a big question that we have to answer because, like I mentioned, it's really important for us to know the mechanism if we want to develop a treatment for this patient. So to actually determine the mechanism of this genetic variant, we decided to apply our transcript sequencing workflow with cyclohexamide. So using RNA sequencing and cyclohexamide, we were able to find that MFN2 actually does undergo nonsense-mediated decay in this patient. And in fact, this actually showed up as a top hit in our genome-wide search. So we looked at the entire genome, all 20,000 genes. We looked at the mRNA for every single gene, and we found that MFN2 showed up as a top hit as being under nonsense-mediated decay. We then looked at the actual transcripts that were sequenced by transcript sequencing, and we see that this patient has a lot of transcripts that have altered RNA splicing and that undergo nonsense-mediated decay. So basically, the findings here confirms the genome-wide finding that we saw here. So I just showed how we use transcript sequencing and a nonsense-mediated decay inhibitor called cyclohexamide to help us identify the genetic cause and mechanism for this patient. So this is really powerful because we confirm the genetic cause for this patient, and we now know the mechanism in which the mutation actually causes disease. So this means that we're able to use this knowledge to actually inform the development of an antisense oligonucleotide treatment for this patient. And so this patient also gave an interview to the American Society of Human Genetics about 1 year ago, talking about the impact of this genetic diagnosis. So if you guys are interested in like a very real world situation and how medical genetics can actually influence lives of patients, I think this is a really cool article to check out. So this brings me to the end of the talk. So today I talked about how my lab is developing this new method to find pathogenic variants and how we applied that to one patient already. And we're going to be applying this, and we're currently applying this to 50 plus patients. And if this works out, then our hope is we can apply this to hundreds, maybe even thousands of patients in the future. So thank you all so much for being here in my talk. I want to thank my lab for all of their support, and happy to take any questions. Thanks.
We could probably take one question before we take a 10-minute break. Go for it.
I guess my question is, with the different variants, I'm just wondering if there's like opportunity for different or more personalized therapeutics, since you're seeing it from a clinical standpoint, now that you have like this deep dive access to their genome and their RNA and their proteins and pathways. Can you use that information to kind of personalize their care?
Yeah, I think that's a really awesome question. And I think that's a question that is kind of foreshadowing what the future of medical genetics is going to look like. So for example, the type of sequencing that we're doing it for every single patient.
Right.
And this genetic cause is specific to this patient.
Right.
And when I talk about antisense oligonucleotide therapy is actually a very personalized treatment.
Right.
So the idea is that that treatment will really work for just this one patient and be very targeted. So we can treat this patient with minimal side effects. So I don't have a slide to show how antisense oligonucleotide therapy works, but essentially for this patient with MFN2, you can imagine, we know that MFN2 is the gene with a problem. So we can develop a antisense oligonucleotide therapy to target just MFN2. Right. So that's an example of how we can use this method to come up with a precision therapeutic.
Yeah.
Yeah.
Very cool.
Yeah, thank you.